but it still holds value in the way that we should think about stress itself. So, is that what we're measuring, what we're interested in, is understanding the effect of unpredictability, right? Sort of measuring or quantifying the effect of something being unpredictable. So, how are we going to approach this? Again, it's a challenging idea here. So, we're comparing a stressor with a small degree of predictability to the same stressor administered in an unpredictable way. Okay? So, in one case, right, this is going to be what we would say is the signaled shock case. Now, obviously, the control case is what it is. It's just a rat in a cage, right? So this gives us a baseline and something to compare our stressor to. So what we're going to do is shock the floor. We are going to electrify the floor and thus shock the rat sitting on this grid. Now, in condition one, we're going to in this case it was play a tone, but you'll see this kind of thing, a light goes on a split second before the shock is administered in the controlled shock or the signaled shock. In the other, the tone and the shock are completely unrelated. Okay? Both animals get the same stressor, right? The same physical shock, the same number of times, and they hear the same number of tones. So the only difference between these two animals with respect to the stressor is that the signaled shock, right? The shock is preceded by a tone, so that the animal knows the shock is coming, versus a tone that is unrelated. So essentially, if you imagine what this is like, so just imagine you were just like walking around randomly and you would get shocked, right? That's basically what this is. You're just walking around, going about your day, and someone's going to deliver a shock. Now that seems pretty terrible, and I agree that it would be, but oddly enough, and perhaps even to a degree counterintuitively, to just know the shock is coming a split second beforehand changes the effect of stress. Now, so how big is that effect? We said it's about 5 times. So, and again, just median length of lesions. This is a kind of an important feature. What are we measuring? Stomach lesions, what do we call it? A stomach lesion is also called what? Ulcers. Okay? So we're measuring the size of ulcers being produced by our stress protocol. So most, right, about 80% in humans are from Helicobacter pylori, right? But they're still, even in that context, there are 20% of these that are stress-induced, right? And that's true in humans. And if you severely stress an animal, right, or even a human, if you put them under severe stressful circumstances, they will themselves develop ulcers under conditions like this, right, where you see a lot of worry. And interestingly, it's where the book Why Zebras Don't Get Ulcers. I don't know if you were here when we were talking about that book. That the whole idea is that overly worrying can actually induce significant physiological harm, right? And this is an example of that. Right now, typically, it's a relatively unusual measure that we haven't really seen before, but that's what it's measuring, a kind of chronic stress, right? And so, what is the takeaway here? What would you say is the takeaway?
Increases stress response.
Yes, increases. Yes, and could you say a little bit louder for everybody?
Unpredictability increases stress response.
Right. So is getting shocked all the time a stressor? Can an animal handle it pretty well if they just get a little bit of a warning?
Yes.
I think that's probably the most interesting part about this, right? So I think both of these groups are, this is not a massive difference between these two groups, right? But especially between control and the unsignaled shock, you have an absolutely extraordinary effect. Right? Okay, so again, this is I think has a very important message for how to deal with stress, right? Again, and this is very true for humans, right? If an unpredictable environment is essentially the most stressful state that you could be in. Even if the environment is terrible, if it's predictable, the effect of stress is less. Even, like I said, even if the environment in a sense is worse, if it's predictable, then that is in fact a little bit better for the, as you can see, the health of the animal. Okay, like I said, we got a couple of little slides. We're going to kind of go through these a little bit more quickly. So part of the, or I should say, notice that stress itself is a relatively, we haven't talked a great deal about what cortisol does, but cortisol is a hyperglycemic hormone. We've mentioned this before that if you have a challenge, you need to throw resources at it. So cortisol is something that frees up those resources for use. And so depending on the species, certainly for seasonal breeders, the breeding season itself tends to show an increase in glucocorticoids. Now, this is typically referred to as allostasis. So you go from one condition to a different type of condition in a predictable way, but that secondary condition needs a lot more energy, right? So the breeding season requires the animal to use more energy, whether that be for fighting for males, parenting, all of these things require much more energy than the non-breeding season does. And so we can actually take a look at differences in the stress response in the breeding versus non-breeding season, and that's kind of what's shown here. So the red line on top, so this is one of the older figures, it's red and green, so if you're red-green colorblind, the line on the top is stress, the bottom line is baseline, and so in the red, you have, notice you've got a much bigger stress response in the breeding season than you have under sort of normal conditions, right? Even regular stress goes up, right? And induced stress is even larger. One other point about birds here, so this is a rather interesting experiment. So essentially they were looking at the effect of stress, right? So cortisol influences how high birds will fly and how high they will roost, right, in sort of bad weather situations, okay? So if you are stressed, how are you migrating? As close to the ground as you possibly can, right? So stress induces, right? Now again, these are birds. We're not stressing these birds. We've quite literally just implanted them with cortisol. So their stress levels, right, their cortisol levels are higher than baseline. And so notice that the sort of normal animals are much higher up than our cortisol implanted animals. Another kind of interesting element of this, we do another port implant, and what we're looking at is the change in range, right? So if we kind of remember way back to our general adaptive syndrome, right? So the sort of generalized adaptive syndrome, that in the resistance phase, right, you need to, if you're starving, what should you be doing? If you're a bird, what should you be doing? Searching farther for food, right? I think this makes sense. If times are good, you just right around the nest. If times are bad, how do you know times are bad? Well, one way that an animal will respond is higher levels of cortisol are indicative of stress. Stress in some species causes them to range much further to look for food. There's lots of effects, and this is kind of one of these that I don't think this is going to surprise anybody. High levels of chronic stress are upstream of a lot of bad things, right? And there's just a couple of examples. Survivors of concentration camps have much worse health outcomes long term. Subway, now this is an interesting one. So this is subway train drivers. Right? If they had an accident that killed people, they were much likely to have major illnesses later on. Right? So I think we can all understand that being a massive chronic stressor, right? The guilt associated with that. Again, notice it's a psychological effect. Right? It's a psychological effect that leads to a physiological sort of manifestation of this. And sort of the original consulate, high blood pressure, muscle tissue damage, infertility, suppressed immune system, diabetes, growth inhibition, all of these things are or can be the result of high glucocorticoids over time. Now, the study that's shown over there, this is air traffic controllers at, right, like you can imagine the top one is like Hartsfield, and the bottom one is, you know, some kind of rural airport, right? They both have air traffic controllers, but you have to imagine that the world's busiest airport, it has to be the worst job, or at least a very high stress job to be an air traffic controller in Atlanta. Okay? And if we kind of look at hypertension by age, notice that as these individuals age, their percentage of hypertension goes up dramatically. I think we can kind of skip over this because again, I want to move on to make sure that we have enough time to cover all the major topics of homeostasis, and those aren't sort of significant figures for our general understanding. They really just reinforce what we've already looked at. So, as a summary, the stress response involves lots of changes in the endocrine and nervous system. Most of these are sort of conserved, meaning no matter what the type of stressor, you're going to get relatively the same response. These are adrenal mediated, so both epinephrine, norepinephrine, and cortisol involve changes in the adrenal glands. Acute stress can be beneficial. Chronic stress is often very, very detrimental or harmful, and the intensity of the stress response is altered by psychological factors. Okay, so these psychological factors, as we have just seen, have a massive impact, right, on stress. Because again, if we think about it from a human perspective, and this is kind of one thing, right? Small stresses are good. This is why we go to the gym. This is why we challenge ourselves. This is the beneficial effect of stress. But notice that, if you're worrying about something, that is a psychological stressor that you are essentially imposing on yourself. Because we all know, does worrying make any difference? No. It actually, and this is a fun fact, it actually makes it worse. Demonstrably, measurably worse to worry about something that you can't control, right? And so, hopefully we can use that, armed with that information, to work on controlling our stress response to psychological stressors, which is a very critical component of health. All right, so we're going to do the same type of thing that we did, that we always do, where we leave the last sort of half hour or so for questions, but so I'm going to go through these pretty quickly. Again, the general pieces of homeostasis, a lot of this stuff I think you're going to have had in other classes, and if you want me to sort of pause and go through something, please just raise your hand and say, I don't really remember that, could you go over that quickly? But I'm going to try to move through it so that we can get to the critical figures, which again I think are going to be ultimately more important. Claude Bernard, Walter Cannon, two of the most famous physiologists and physicians that sort of in the sort of modern era, they were critical to developing this idea of homeostasis. I think we have, right, we had our endocrine crash course, we kind of have an idea of what homeostasis is, right? So I think we can kind of move towards a kind of direct to the figures. Couple of key features that you need a set point. So the system is trying to maintain a set point. We're going to be focused exclusively when it comes to homeostasis on negative feedback homeostasis, right? So essentially the idea is the system is trying to maintain some set point. That can be for blood sugar, blood oxygen, blood CO2, right? pH, anything, right? Anything and everything. Neuron activity, right? Ventilation rate, right? All of these things have set points. And so basically, in order to, if you have a set point, you need a sensor, right? So you have to compare what the value should be to what the value is. So you have to have some way to know what the value is, and then you have to have some levers to pull, right? To change the value if it's not within the range of the set point, right? I think we're all, like I said, this is negative feedback 101. Now, this is a great experimental design, okay? So, notice that your body generates fevers, right? So I'm assuming you guys all know this, right? This is not some revelation, right? The fever is not caused by the pathogen. The fever is caused by the immune system, right? The immune system turns up the internal temperature, right? So the system moves the set point, right? Now, the question is, does that do any good? You guys are kinda, right? Maybe. We'd like to hope so, right? I mean, the fevers are not pleasant, right? So notice that the sort of evolutionary logic that we apply to all living systems should say that, well, it probably does something important, or else it would have been eliminated, right? Now, the determining whether or not that's true is challenging, right? Because essentially notice that if you, right, if we try to adjust our own set points down, what is the system going to do? It's going to bring it back up again. So if we want to know whether or not a fever, right, is a net positive for the organism, it's almost impossible to ask this question in mammals, right, or any endotherm. We can include birds as well, because the system is controlling the temperature, okay? So what this is asking is really a question in two parts, right? First, if we have an ectotherm like our iguana here, or really any lizard, right? So an ectotherm is not able internally to regulate its own body temperature, but it can regulate its own body temperature behaviorally, right? Now, to kind of understand the way that this works, you have an iguana in a terrarium there, right? And there's a heat lamp on one end of the terrarium. If the lizard wants to be warmer, it goes to the heat lamp. If it wants to be colder, it goes away. So behaviorally, it can set whatever it wants to be at. Now, so the first question, in order to answer our real question, is we have to know if we infect an iguana with something, will it induce a fever behaviorally? Right? That's the first question. Because if it doesn't, if the iguana doesn't induce a fever, then there's really like no point in part two. Yes?
Is it a pathogen?
So we're injecting the sort of same kind of, dead bacteria in the first one, right? So notice like the sort of classic immune, if you want to test immune response, you put in a dead pathogen because you can essentially measure independent of the effect of the pathogen. Like if you inject something live, right, there's lots of other variables, but if you inject something dead, really all you're measuring, you're injecting a standard amount of essentially like dead bacteria that's going to induce an immune response of a certain size, right? And so what we're going to do to our iguanas is we're going to inject them with a bacteria. Right? In this case, this bacteria is dead. Right? Part A, it's a dead bacteria because what we want to know is, will the iguana move to the light? Right? Will they induce a fever? So if we're looking up here, day 1 is in green. So how warm does an iguana want to be in the middle of the day?
Around 36.
Yeah, let's say 36, right? 36 Celsius, right? Centigrade, okay? So that's our baseline. The animal is just like living its normal life. It likes to be warm in the day, right? About 36, okay? Now, we're going to then inject the bacteria. This is the red line. Do the iguanas behaviorally induce a fever? Yes. So what temperature do they get to? Yeah, above 40, right? Maybe 41, 40, 41 degrees. That's significantly warmer, right? That's what it looks like, about 4 or 5 degrees Celsius, right? So notice that if we were at 98 and, right, you're getting a fever at about 103, right? That's about the same. It's close. It's actually a little bit more here, but, this is a significant thing. Now, because we see that it's dead bacteria, what happens on day 3? Well, the immune response goes down, right? The sort of pathogen has passed, right? And it goes back down to its normal level. So we have, again, notice what we've done here. We've demonstrated that an iguana will do behaviorally the same thing that a mammal will do endogenously, sort of naturally, right? Internally, I guess would be a better way to say it, right? Everyone understands that? So an iguana will behaviorally do the thing that we do naturally. So that allows us to ask the question, right? We're going to take advantage of the fact that we can control how warm they can be. Now, how do we do that? We can essentially put them in an incubator at a certain temperature, right? Notice that if you set an incubator, you're setting the internal temperature for the lizard, okay? So, now what we're going to do is instead of, now this is going to be part C, right? Remember, what's the question we're asking? Yeah, is a fever a net positive, right? I think that's an easy way to say it. Then you can remember this study the next time you guys are sick, right? Hopefully that's not anytime soon, but you remember that. The question we're asking is a fever a net positive? Okay, so we know now that an iguana will induce behaviorally a fever. So what we're going to do in part 2 is answer this question. So we're going to inject our iguanas with live pathogens. We're going to inject them with live pathogens, and then we are going to put them at different temperatures. So there's an incubator at 34, an incubator at 36, an incubator at 38. Now these are different iguanas, right? Different groups of iguanas in each of these incubators, right? So each one is a group, all right? Now, what are we measuring? How long they survive. So every day, we're basically going in there and checking to see if they're not dead. Right? That's the idea. We're gonna, every day we go in, oh no, this one's dead, we can pull it out. Right? Okay, so what's the normal body temperature of an iguana? 36. So let's go down to 36. What is the week-long survival rate at 36 degrees? 20%. So 8 out of 10 of our iguanas are dead. How is it at 7? How about if at 34 degrees? They're all dead, right? They're all dead at 34 degrees. 80% of them are dead at 36 and 38 degrees, right? What about the warmer temperatures? We said we were gonna move them up to around 40 to 41 degrees, right? That's where the iguana's like sweet spot is, right? That's where they made themselves. So, what's the effect of that? Much higher survival rate, right? 70, 75%-ish, right? So 7 out of 10 iguanas survive at high temperatures. You know, at cold temperatures, none of them survive. Right? So, just so we're summarizing here, what are the critical comparisons? Well, yes, okay, go ahead. Well, that's the question, right? But we want to say what the critical comparison, how do we, what pieces of data, and again, what pieces of data show the answer to the question? Okay,
Would it be the ones who were placed in 42 and 43s have a much higher survival rate than the ones placed at 20 and 36?
Right, so the way that I think we could get to, right, so notice what our question was, which I thought you did a great job encapsulating what the question was, right? Is a fever a net positive? So how do we answer that question? Now we've gotten all the way to the point where we're taking the data. So what tells us the answer? Well, animals incubated at fever-like or high temperatures survived at about 75% of a rate, and animals at cold temperatures were 20% or lower, right? So notice how, if I said, are fevers beneficial? I said, well, how did you test it? Well, we put animals at specific temperatures and asked if they lived. The ones at high temperatures lived, and the ones at cold temperatures died, right? So is a fever a net benefit for, yes, right? And so notice again, I'm just kind of harping on this critical comparison issue because if there's so a lot of times it's when your answers are sort of in the 7, 6, 7, 8 range, it's often the critical comparisons, right? So again, notice when we had our question, what on here answers that question? High temperatures live, cold temperatures die, right? I mean, we can certainly be more precise than that, but that's essentially the idea. Okay. We've got to talk a little bit about salt and water, okay? So what we're going to be talking about is basically what amounts to dehydration of different types, okay? So we've got really two types. They're named after like types of thirst, but really they're types of issues with water and salt balance and blood pressure. So if we think about the organism, where's most of your water? In cells. How much of it's in cells? Looks like 67% from here, right? 67% of all of your water is in cells. How much is in the blood? 7%, so not a lot, right? Right, not proportionally not a lot. And so the interstitial fluid, right, has even more, right? 18% or 26%, and then the blood has low. Now, I don't know if do I need to review osmosis? We're good. Okay, great. Let's go. Okay, so we get the idea. So I think you're gonna get this idea pretty quickly. So there's really kind of two major types of thirst. We're gonna call them osmotic thirst and hypovolemic thirst. So this is kind of like, if we just remember the names, I think, and we think about what that means, we should be fine, right? Osmotic means that you're essentially going to be pulling water where you don't want it to go, right? This is if we were sitting here and just eating like salty potato chips or salty pretzels or something, notice that salt is going to be pulling water out of your cells and towards your blood as your blood is more salty, right? This is osmotic thirst. So extracellular high osmolarity in the blood plasma, typically, but potentially also in right? The extracellular compartment more generally. Hypovolemic thirst, hypo meaning under, volemic meaning volume, so this is a low volume problem. Right? Now, this is the one that's caused by hemorrhaging, diarrhea, excessive perspiration, these types of things. Now, these types of things have different solutions physiologically. So, osmotic thirst, how do you alleviate osmotic thirst? Drink water, right? Drink water. How do you alleviate hypovolemic thirst? Okay. We felt less confident. I loved your confidence on that one. We were like, osmotic thirst, drink water. Excellent. What about hypovolemic thirst? Is water going to solve the problem? No, it's not going to solve the problem. So what is going to solve the problem? Eat. Well, in a sense, right, we have, you must have seen like sports drinks at some point in your lives, right? There, right? And so remember, those things are bad for you unless you're in sort of hypovolemic thirst. But if you, right, are an athlete who is excessively sweating, notice that the problem is not that you're just losing water, it's that you're losing these electrolytes as well. So if you are clinically hypovolemic, this is where they put an IV with normal saline in there, right? So notice that just doing the water thing is not going to work. You need more salts, and with these beverages, right, there's also sugars in there which are osmotically active, and so these are things that are going to sort of restore this balance, right? So if you essentially just have osmotic thirst, water is the best thing. Now, will, let's say, Gatorade solve osmotic thirst? No. Are you still going to be thirsty after drinking your Gatorade? Yes, right? Because you have not changed the underlying problem. Okay, hopefully everybody sees that. Now we're going to kind of come back to these, or we'll talk about them a little bit individually. So, this is a time series. This is a rodent, right? I believe this is a rodent. Essentially what we're going to do, it doesn't really matter if it's a rodent though, it could be really anything, the effect is going to be more or less the same. So I'm going to inject you with hypertonic saline, right? Now, your physiology is going to respond to this. So what are you going to do? If I inject you with salt, what are you going to do? Well, so yes, like that's the initial thing, but notice that your body, your physiology is going to respond to that. You're not going to take that lying down, right? You're going to respond physiologically to undehydrate yourself, right? So what are you going to do? Well, you're going to drink water, and you're also going to do what? Use the restroom? No. The opposite of using the restroom is reducing urine volume. Okay? So so the red line here, this line is your water intake, this is urine output. So notice that the hormonal mechanism that you're going to pull, and so even if you didn't have water around, what you're going to do is release AVP, right? This is vasopressin. Vasopressin targets the kidney and it pulls more water, which is why your urine volume goes down. Okay? So your water consumption, if available, is going to go up. Your urine volume is going to go down because of the endocrine effect, right? So essentially you have in your hypothalamus osmoreceptors, which are measuring the osmolarity of your blood. So if you eat pretzels, what happens? Like we're just saying salty pretzels, right? I don't know anybody who'd eat non-salty pretzels, but it's okay. What happens to your blood osmolarity when you eat a bag of pretzels or potato chips? It goes up, right? Your osmolarity goes up, right? This is another way of saying your blood is too salty now. So the system will respond, right? Obviously, you notice that if you eat a bunch of salty snacks, you're feeling like, oh, I need to drink some water, right? I'm thirsty now. But you also have the release of AVP, which causes greater absorption of water. Now, water, and or in many cases, depending on how big of an effect, AVP is perfectly able to do this, right? So one of the ways that you can sort of tell if you're dehydrated is that your urine gets darker, right? The darkness of your urine is directly proportional to the amount of circulating AVP in the system. So the higher the AVP, the darker your urine. Now, hypovolemic thirst is a much more significant problem, right? So low blood pressure is a very serious issue, right? And it results from water and salt being lost together. So water consumption does not fix this problem, right? This is why if you are severely dehydrated, just drinking water will not help, right? Or the effect of just drinking water in severe dehydration is minimal, right? This is why severe dehydration typically requires like IV fluids, right? And that's true for you as it's true for your pet. There's a point at which you cannot overcome how bad the situation has gotten. So again, and the problem becomes a blood pressure related issue. Now, we need salt and we need water to restore this necessary balance. So our key metrics here are, we are going to need to detect the, right, so this is homeostasis, so we have a set point. We have a set point for pressure, right? What is measuring your set point for pressure are what are known as baroreceptors, right? So these are pressure receptors. When your pressure, right, now again, where can we measure this pressure? Where's a good place to measure blood pressure? Kidneys are a great place. Where else? Arteries, but where should blood pressure be the highest? What artery has the highest blood pressure?
Aorta.
The aorta, right? So if you're gonna watch, so again, notice that this is an effect size situation. So arteries have higher pressure, so it's easier to measure a drop in pressure at places where pressure is the highest. You guys said kidneys, right? Your glomeruli are really super high blood pressure. So any drop in blood pressure is detectable very easy, right? Whereas a drop in pressure in a low pressure, like say veins, for example, is much more difficult to measure. So you want to measure blood pressure where pressure is the highest. Yes?
When taking blood pressure, do you put the device on the
Because you can't really put it on the aorta? Right, but you can, but so the cuff, you're talking about the cuff, because essentially, notice again, remember that with the cuff, you're actually constricting the blood flow and measuring it there. So I think it's really more of a practical thing, right? Rather than like, so notice the body has the advantage of having the sensors on the inside, but you're still measuring an artery. It's the easiest, probably the most, I imagine, now I've never, I would say from a predictable perspective, I imagine you could do it on the leg too, right? Same exact sort of situation. It's just easier, I think, and more convenient to have it on someone's arm, right? And again, notice you have to like pull up the clothing and do all of that kind of stuff. So, but yeah, that's a pressure cuff that then that you're measuring the difference between the high value and the low value there. So I think that's a practical concern, but it's not necessarily going to be as accurate as an internal metric, I would say. I can't be sure on that, they're probably pretty close, but our baroreceptors are essentially, right, so notice that when the aorta has high pressure, it's stretched, right? When blood pressure drops, right, the stretched receptors contract, right? And so this, right, the activation of these stretch receptors are going to, amongst other things here, they are going to cause the kidney to release renin. right? So I'm sure everybody, I teach endocrinology too, so everybody's tired of the angiotensin II system, but we're just gonna go over it quickly, right? I know, I can see the faces, but so it's just review. So again, as quickly as a review for everybody, I don't wanna spend a ton of time on it, but we're going to have complementary things that increase blood pressure, right? It's really that straightforward. So, to give you an idea, one of the major players, we were just talking about vasopressins, probably a little bit out of order, how I would have liked to discuss it. But vasopressin, again, this is your water absorption in the kidney, right? So, one of the other things, and kind of one of the themes of this lecture, is that your hormones control lots of these aspects related to homeostasis, right? So these are things like how much salt you want to eat, right? So notice that sometimes you want to eat a lot of salt, other times not. Sometimes you're thirsty, sometimes you're not, right? And how much water you consume is actually dependent on vasopressin. Or I should say largely dependent on vasopressin. So I see a lot of water bottles in here, right? Lots of them, that's good, right? Important to stay hydrated, agree. How much water do you drink a day? 10 bottles? That's a lot. You must get the cases from the store, right? That's 10 bottles of water, okay. How much, what kind of fluid? Like a little bottle like that?
Yeah.
A 10 bottle, okay. So how much is that, like 12 ounces, 16 ounces?
That's like 16.
That's a 16 ounces. So so 16 ounces, so it's like what, 80 ounces of water a day, right? 160, 10, that's 160 ounces of water. So how much is that? How many, is that a gallon?
That's
So
over a gallon. A gallon's 1/3.
130, yeah, I was gonna say, that's about a gallon. That's
Really
you
good.
like to stay hydrated, that's good.
Absolutely.
I think that's a little over the average.
Right. Normal person maybe feels like their jug of maybe like 12.
Yeah, I mean, I think half a gallon of water is probably a reasonable amount in a day. I think that's probably a sustainable amount, but which is we're gonna blow your mind here on this next slide here. Okay, so we're drinking over a gallon a day.
Maybe like 5 times a day.
So, as I said, so remember, here's our main concern here. What we're looking at is the way that circulating vasopressin, what we're interested in is how vasopressin controls water intake. And how are we going to look at how vasopressin controls water intake is we're going to compare animals that have vasopressin versus ones that do not have vasopressin. So there's a strain of rats called Brattleboro rats. Brattleboro rats, right, do not make vasopressin. Right? This is kind of a, like if you've heard of the condition diabetes insipidus, right? This is another vasopressin mediated condition where you do not have sufficient vasopressin. And so,
So it's not a species, it's a disease?
it's a strain. Brattleboro
Yeah.
is a strain of rats. So it's a lineage, it's a genetic strain that has a specific mutation in the actual vasopressin gene itself, right?
So they're both the same species, but one's a different strain?
Yes,
Cool.
okay?
They
So-
don't make vasopressin.
They do not, right? So, Brattleboro rats do not, Brattleboro, whatever they're called, doesn't matter.
B-Rex.
So, these rats don't make vasopressin. Now, if you were a Brattleboro rat, how much were you gonna drink? A lot? How much? So if you're drinking a gallon of water a day, we'll just take it easy. If you don't have vasopressin and you drink a gallon of water a day, how much water are you drinking now? 10 gallons. 10 gallons of water, right? So this is a 10x difference. You know, so if you're drinking a half a gallon of water a day, right, that's 5 gallons of water. And so this is another way to kind of think about this is that this vasopressin is regulating how much you have to drink. Now I won't go through a lot of the sort of brain regions here. I just want to point out the one major one that's associated with this because it's going to be related to angiotensin II, which is the subfornical organ. Right? So The subcortical organ is the sort of center of thirst in the brain. And if we look at an MRI, notice that, so if we, I know there's going to be a slight issue of scale, but you can see where the subcortical organ is. This is a rat brain. You can sort of tell by the general structure here. But notice that that orange dot is equivalent to that, you see that bright red dot there in the center? That is your subcortical organ. This is a condition of maximum thirst. Notice that after we were drinking water, that the area is no longer active, right? So when this area, right, lights up, the individual has a very powerful thirst, right? So thirst, like most other things, this is something that exists in the brain, right? Okay, I want to go quickly through this angiotensin thing that everybody hates, but I'll let you review this yourself. But this is this is the part that we want to, just to kind of remind us of how this works. So angiotensin 1, ACE, converts it, angiotensin converting enzyme, into angiotensin 2. Angiotensin 2 is the active molecule, right? This is the thing that is going to do the 4 complementary effects, right? 4 things that all increase blood pressure. The easiest one is vasoconstriction, right? Really straightforward. I think everybody understands how vasoconstriction is going to increase blood pressure. Now, and we'll see that far line on the side to the subcortical organ. You are going to have a powerful thirst, right? I think that also, we just covered how that works. But the 2 that are important to sort of focus on for us are vasopressin, which I think we just reviewed what it does, right? It causes water absorption in the distal tubule and collecting duct in the nephron, right? So you're just absorbing more water. And the more water you absorb, the more concentrated your urine becomes, right? So that's because you are moving water from the kidney, right, from the tubule into the blood. But we can actually do better than that. And remember also that our issue was that when in conditions of hypovolemic thirst, we don't need just water, we need water and salt. So we've got to get some salt in there too, and that's where aldosterone comes in. Aldosterone is a mineralocorticoid, right? And we're going to spend a little bit of time talking about those mineralocorticoids now, okay? So notice there's another really important factor here. So aldosterone causes salt to be put into the blood. Notice that that's also going to increase the movement of water into the blood, right? Again, essentially increasing the saltiness of the blood is going to make vasopressin and sort of the aquaporins that vasopressin causes to be inserted to absorb more water. So these things are complementary. Right? Everyone has heard of aldosterone before or no? Yes? I have some yeses, some no. All right, so we're gonna use it in the context of a couple. We're gonna use it in one ecological context and sort of one experimental context. Essentially, it responds to angiotensin. It stimulates a sodium pump, right? So salt is pumped into the blood, right? It's obtained from diets. It's maintained in the interstitial fluid. Sodium, obviously very important stuff. So let's look at aldosterone. And so this is one that requires a little bit of explanation. Right? So essentially, DACA, right, this is deoxycorticosterone acetate. If I inject this into any mammal, that mammal is going to consume a lot of salt, right? It's a known pharmacological effect. I inject you with this, right? If I injected you, or a rat, or a rabbit, or really anything, I inject you with this drug, and you will consume more salt. Right? So notice with our sort of normal condition here, we go from about 1 milliliter of salt water to 6 milliliters of salt water. So that's what, 12 times the amount of salt consumed in a day. So from that baseline, what we're asking, the essential question is that salt consumption mediated by the mineralocorticoid receptor? Okay? So we know the effect is known before this, right? So we know if we give animals this drug, they're going to consume a lot of salt. Yes?
Wait, is that salt consumption mediated by the
Mineralocorticoid receptor. So aldosterone is a mineralocorticoid, right? So you know how we talk about cortisol or corticosterone being a glucocorticoid, all right? So these are both things that are made by the adrenal cortex. So glucocorticoids are made, mineralocorticoids are made, and so the mineralocorticoid, the main mineralocorticoid, is aldosterone. And notice that aldosterone itself is what causes salt absorption, right? So our question is, if when we inject this drug, right, and we generate this massive salt consumption, we want to know if that's mediated by the aldosterone receptor, essentially. But it's just called a mineralocorticoid receptor, right? But it's really the receptor that aldosterone binds to. Now, why might we think that that's the case? Well, we know that if we give aldosterone, right, we're going to take up more salt. So the way that we're going to do this particular experiment is we're going to say, okay, well, we have this pharmacological effect of salt, right? Salt consumption. So we're going to essentially try to block the mineralocorticoid receptor. Now, in some cases, we've seen, well, we could just use an antagonist, right? You inject a drug, it binds to the receptor, and that's good. But that only works if you have a good and specific drug for that particular receptor. And in this case, we don't have that. So we're going to use a little bit more of an advanced technique, which is an antisense oligonucleotide. So because receptors are proteins, they have to go from mRNA to protein. So what we can take advantage of that sort of central dogma and throw an antisense oligonucleotide in there that has a specific sequence that we know will complementarily bind to the mRNA of our mineralocorticoid receptor. Okay? Now, what do cells do with double-stranded RNA?
Make protein. Oh no, it's blue.
I mean, I took you to like always taking these like virology classes, and like you guys are like taking these classes like all over the place, right? Like this is a very viral department, and so what do cells do with double-stranded RNA?
Make protein.
Yeah, yes, it degrades. Why does it degrade?
I don't know.
What is the only time you're going to find double-stranded RNA in a cell? If you're a eukaryotic cell, when's the only time you find double-stranded RNA inside of yourself?
Viral infection.
When you've been infected with a virus. So why do cells care about double-stranded RNA?
It's foreign.
It's foreign, and they're going to chop it up. So if you chop up all the RNA, what happens to the protein?
No protein.
It doesn't get made,
Right.
right?
I guess.
That makes sense?
For sure.
Your virology teachers are going to be very disappointed, right? It's part of the sort of cells like ancient, right, defense against viruses. So double-stranded DNA, great. Double-stranded RNA, let's get rid of that thing right away. So as soon as things form double-stranded RNA, you cleave them, you just chop them up. You do not want them any longer. And we, being the clever primates that we are, figured this out and then realized that we could use this to temporarily knock down genes of interest. Okay? So this is what MR antisense is, the mineralocorticoid antisense, right? And so this is, you can think of this, right, as a temporary knockout of the mineralocorticoid receptor. The control is an antisense oligonucleotide that's scrambled up, and so it doesn't bind to anything, or at least it doesn't bind to the mineralocorticoid receptor. Right? So it's kind of like a sham almost, right? So you're putting it in there to control for introducing this kind of genetic material into your experiment. Now, is this effect mediated by the mineralocorticoid receptor?
Yes.
Yes. We can say yes. Now, who wants to do the critical comparison? How do we know? You said yes, you were right. Somebody said yes, they're right. It is, the answer is yes. So how do we know? How would you tell somebody that we knew?
Given that temporary knockout reduces salt intake, while the sham still has a little bit of the salt intake, like the control, without any knockout to anything.
Okay.
I have to fix that.
All right, so we'll work on the phrasing, right? So so the idea is we're looking at what column, what are the two most important columns to compare? Antisense, which one? Is it are red and blue equal to? Which one's the important one? Red or blue? Blue. The blue, this one, to what? You either pick this one or this one, right? Because the effect is the same. Notice that this is what we would call a technical control. So if this one's significantly different than this one, we have a problem with our technique, right? If this one is significantly different from this one, but not this one, we have an effect. Does that make sense? So our comparison, our critical comparison, so again, notice what we're doing. We're formalizing we can look and say, yeah, there's definitely an effect there. But notice for our explanatory purposes, we want to talk about how we can be confident in saying yes. Because the MR antisense gives you consume about, 1 milliliter versus 6, right? That's a 5x difference in salt consumption, right? And that tells us in a kind of simple way, yes, the mineralocorticoid receptor is responsible, because if we block it, we block the increase in salt consumption as evidenced in our critical comparison between our antisense DACA compared to our saline DACA, right? Yeah? Okay. And again, notice, I know I'm harping on this, but we're coming up on the exam, and I really want to emphasize that what a critical comparison is the, right, column A, the important column here compared to the important column here, the important line here compared to the important line here. That allows us to extract the important information. Sticking with our aldosterone theme, right, we are going to be looking at more of an ecological effect here. And so rabbits are not native to Australia, but they are everywhere, right? And so that means that when rabbits were brought over, they diversified their habitats. And so Australia is a place where we have some habitats that are very high in salt, and some habitats that are very low in salt. Okay? So, where is the highest salt found? In the desert. Very salty out there. Low salt in the mountains. Okay, now what we're looking at are, notice here, physiological adaptations to essentially assist with homeostasis, and again, assist with adapting to a new environment. So what do the rabbits that live in the snowy mountains do? How do they adapt to the lack of salt in their diet? They produce a lot more aldosterone, right? So notice, if we're comparing high salt to low salt, that we've got, basically 6 times the amount of salt, over 6 times the amount of aldosterone produced where salt is low. compared to where salt is high. The adrenal mass doesn't change that much, but notice the composition does. So there are, and we didn't talk about this, it's a little bit less important for our general idea, but the zona glomerulosa is the layer that makes aldosterone. If that wasn't probably, you can probably intuit that that was true, right, just from looking at the table. So the relative percentage of the adrenal cortex that makes aldosterone increases when there's no salt, and essentially, right, just doesn't produce very much aldosterone in high salt environments. Now, all right. This is kind of like a biophysics experiment, and what we're really asking here is, can so let me throw this back to the idea of the iguana. The iguana could not internally regulate its temperature, so what did it do instead? It behaviorally regulated its temperature. Okay, right? So because it could not do it itself, it used behavior to regulate, right? Now, what we're asking is whether or not, right, we can do the same thing with salt content. Okay? Now, if you adrenalectomize a rat in this context, what do you lose in the context of salt? Aldosterone. Yes, aldosterone. Now, so notice that our rats cannot do, right? They cannot do what our rabbits did because they do not have the ability, right? We have cut their adrenal glands. That's the ADX. Okay? So we are going to compare. Now, notice we're going to compare normal rats, i.e. with intact adrenal glands, and adrenalectomized, and we're going to compare their preference for salt at different concentrations. All right? Now, if you're going to run a preference test, how do you do it?
No.
If I asked you to, right? So if you wanted to design this experiment, what do you have to give the rats?
3-chamber test.
We could give them a 3-chamber, but we'll put it all in one chamber. We're gonna give them 2 things to drink out of.
Okay.
Right? One has salt water of some concentration in it, and the other has plain water. Okay? So if you have a low, right? So what does no preference mean? Equal, right? You drink as much of the salt as you drink of the regular water. Okay? So if we're just talking about normal rats, at what concentration of salt do normal rats prefer salt? What's the starting concentration?
2. Oh, the concentration. Yeah. 2 should be good.
I mean, the range of numbers you guys threw out there.
So,
Yeah, it's a little bit, you're saying 2.
yeah.
Right, you say the question again.
A great stalling tactic. I'm going to need you to repeat that question real quick here. What concentration of salt do normal rats begin preferring salt?
I don't know.
It starts going up at,
Preference. Yeah, 1.
right
Yeah.
after 1.
Yeah.
Okay.
Like 0.5 or some shit.
So, again, the reason I ask this is because if you're probably not used to looking at figures like this, so it takes a second to orient yourself. So, concentration is on the x-axis. And yes, it is handily labeled there, preference threshold, which what we want to do is put a number to that, which is what? What should we put a number to?
1.8.
27.
I meant 0.8.
Okay.
0.8.
Yeah, sure, 0.8, fine. 0.8. What concentration is aversive? 2.8, right? So notice this is a percent solution. So essentially 0.8% salt, you will drink more. Now notice that, remember we said no preference means 50-50, right? So anything above 50% suggests a preference. Anything below 50% is an aversion, right? That kind of makes sense, everybody? All right. So now,
Wait, that don't make sense because the arrows before it goes under 50%.
it's at the dot there. It's at the line. Okay? It's at the line. This is the aversion threshold. Below this, it's aversive. Above it, it's preferred. Right? You see that?
I would think
Where-
it would intersect, but I'll take your word for it.
Where it goes below the line,
Mhm.
it's aversive. Right?
Yeah.
Where it's above the line, the 50% threshold, it's preferred.
What is aversion?
Yeah, it makes sense. It's just not what it shows. Like, I get that.
Okay, the aversion threshold. Okay, so if you have no preference, half of the water half of what you consume is gonna be bottle A, half of what you consume is gonna be bottle B. When you start preferring, right, now notice that bottle A is a neutral stimulus. It's literally water. Okay? So I'm gonna keep adding salt to bottle B. You can do this at your house, right? You can keep adding salt to the water, stirring it up, and at some point you won't want to drink it anymore. At some point you'll prefer to drink it, and at some point you will take the regular water over it. Okay? So aversion threshold means it's no longer 50-50 for the test substance, and it's below, right? So at that case, like, so at 3%, right, at 3% saline,
Is it
I'm,
soluble?
you're not drinking very much of it, right? You're drinking a lot more water. You're drinking about probably 60%, 65% water, and like 35% of saline. And now notice that we don't even get to 4, right? It's gonna be basically at 0. You don't, at 4% saline, that's like drinking ocean water, more salty actually, right? So you're not gonna wanna do that. And again, so what we're trying to do here is we're gonna slowly increase the concentration of salt, and we're going to measure their preference or aversion to that thing. Right? Now, remember, we gotta sort of armed with that knowledge, it allows us to make the comparison between normals. Or
So, at the preference threshold going forward, there's no preference. And then until you get to the aversion threshold.
So you said there's no preference. So starting when the line is above 50%, it means you prefer it. Now the strength of your preference is what is essentially being measured. So if we take the peak here, right? So it's roughly, let's say it's about right here, so maybe like 1.75. Notice that at 1.75% salt, right? You picture a rat with the little things coming out there. At 1.75% salt, 80% of your drinking is the salt water. Okay? Ish, right? If I'm just saying the peak is at 80%, right? There's not a lot of scale bars there, but it's about 80, right? Ish. So 80%, that means you're still drinking some of the regular water, but you, right, to 80-20 prefer salt to, right? Yeah? Okay. Now, there's another line on here that says the detection threshold. So the detection threshold is essentially, if I keep giving you salt, remember I'm putting a little bit of salt in here and I'm giving you 2 glasses and I'm asking you, which one has salt in it? And where you can tell that one has salt and one doesn't is what we would call the detection threshold, right? It's the ability of the animal to tell whether there's salt in there or not. Because below that, if they can't taste the salt, it's very hard to have a preference for something that you can't taste, right? Now, our adrenalectomized rats are going to be behaviorally compensating for the fact that they don't have enough salt. Right now, why don't they have enough salt?
They don't have
Aldosterone, which means they cannot absorb salt efficiently. It doesn't mean they can't absorb any salt, but they can't pump it actively. So they have to rely on essentially what amounts to diffusion, and you're not getting very much salt through diffusion, right? This salt is used by active transport. That's what aldosterone does. So essentially what we are doing is, right, how soon do these rats prefer salt? As soon as they can taste that it's in there. And how much do they prefer it? Yeah, like pretty close to 100%. All the way out to 4% saline, which is massively aversive. It would be like literally drinking salt water, right? Ocean water. They are sucking that down like crazy. Now, notice that they're doing that because they can't get the salt in, right? They are salt deprived, right? So if you drank salt water, it would be dehydrating because you can efficiently absorb it, but they cannot. So they have to take in huge amounts of salt. Okay? So are they behaviorally able to compensate like our iguanas for a lack of internal body temperature? Yes, they are. Okay. So I think we're going to end there. For today, we've got a few more slides for energy balance. There's a couple of critical ones, but we'll be able to go through those pretty readily. We'll have to probably cut out some stuff, which we'll talk about then. But I don't want to overload you with like 2 days left. But this is a good portion of homeostasis, right? We've talked about a number of really kind of important figures here. We have our vertebrate rats, we have our iguanas, we have our taste preference, right, in our adrenalectomized animals. We have our hypovolemic thirst and our osmotic thirst. We've got vasopressin, aldosterone, right? These are, yes?
Sorry, with the ADX, is it because they can't absorb salt or
They can't absorb it very efficiently, right? So the problem, aldosterone can't make salt, like it can't make salt, right? But what it does do is cause salt that you've already taken in, right? Things that you've eaten and stuff like that, to be put into the blood actively, right? So what aldosterone does is essentially runs a sodium pump, right? So if you can't run that pump, you can't take in that salt very efficiently. It's not that you can't take in any, but it's the efficiency of the interaction, right? So I want to spend the last 30 minutes on questions again, because this is really our sort of last, but we'll do the same thing on Friday. Maybe we'll have a little bit more, or on Tuesday, maybe we'll have a little bit more time. But again, I can't emphasize this enough. You don't want to be starting studying on Tuesday after class, right? That's right. We want to be preparing, and I know probably many of you are. I'm just trying to things are going to come fast and furious here, so we want to make sure that we're preparing so that if there is something that you really don't understand, that we can make sure to go over it, right? And get to that point. So, does anybody have a figure they want to talk about? We're doing a kind of open review here. You pick a figure, talk about it.
Prenatal stress.
Prenatal stress. Okay. Is there a particular prenatal stress one that you want?
The
Like-
adrenal and.
Like this one? Okay. All right, so, just so we're clear, I think it's always important to note what we're measuring. What are we measuring?
Cortisol.
We're measuring cortisol. In what context are we measuring cortisol? As an adult, after stress, right? So we are subjecting adults to a stressor, and we are measuring the cortisol response. Okay? All right. So since you asked the question, I'll ask you, what are we interested in here?
Well, I have here that whether stress in the womb affects the adult in significant ways.
Okay, so we don't like insignificant ways because you notice again that we're always we have something specific that we're interested in, right? So that's why I asked, what are we really measuring? What would we? Yes, we're measuring cortisol. Yes, we're measuring after stress, right? What do we call that phenomenon? Yes. Stress reactivity. So so that's what we're measuring. We're measuring stress reactivity of the adult, and we're comparing whether or not that adult's mom, was stressed in the womb or not. Right? So can anybody formulate the essential question here for us?
Does prenatal stress affect adult stress reactivity?
Sure, sounds good. I like it. Right? And again, with all of these, there's a number of ways that we can formulate it, and it's important for us to, it's critical to phrase it in your own words, right? Because those stick better, right? Whatever you use to describe it, that's fine. Okay, so we're interested in whether or not prenatal stress affects adult stress reactivity. Sounds good. I like it. So how are we going to measure this? So remember, part one is what we're doing, and then part two is how we're doing it. How are we going to do this?
Looking at stress and non-stress.
Right? We're going to look at the offspring of stress versus non-stress mothers, and we're going to measure
Their stress response.
yes, their stress response. What do you say? Their cortisol response to a stressor. Right? Great. I like it. Okay, so what is our critical comparison? How do we know? Remember, we're trying to answer, we're doing this experiment to answer a question that we just articulated. Does prenatal stress, right, affect adult stress reactivity? So in order to answer that question, we have data. What is our data? What is the critical comparison?
The rats with the stressed mother have a higher stress response,
They do, compared to,
a non-stressed mother.
okay, and so we can do the extra great version of this, right? So that was a good version. Let's do a great version. What should we be adding to that to make it a great version?
Without the adrenal.
Well, no, numbers.
It's
Oh.
triple.
Numbers. Right? Again, I said that was a good one, right? But we want to, right, if possible, always add numbers. Right? Okay, so what is the difference?
It's like triple stress moms, triple stress reactivity in adults.
Okay, great, right? So adults, the adult children of stressed moms have triple the stress reactivity as do the offspring of non-stressed moms. All right, yes? Right, this is our question. This is our critical comparison, and where possible, add numbers. Okay, now, there's another component here, which is important, right? Now, what is the second part about?
If it's hormone mediated.
Okay, ooh, oh, great. There's lots of hormones. Can we be more specific?
Adrenal.
Okay, great, all right. Is the effect that we noted, right? The 3x effect on stress reactivity, is that dependent on adrenal hormones? Did you have a complimentary question over here? Actually, you had your hand up. I just wanted to-
Oh, it was just like, it was that.
Okay, great. So, to do that, to answer that question, what are we gonna do?
I'm gonna look at the other side of the chart and compare
We're gonna compare,
adrenal intact and adrenal ectomized mother rats.
right, okay. So to answer the question of whether or not, right, the hormones of the adrenal gland are the factor that is changing the stress response, i.e. is mom's stress response affecting the offspring directly, we are going to use adrenalectomized moms and do the same thing, right? Does it turn out that adrenal hormones are the key?
Yes.
Yes. And how do we know that? I don't like that. The stress changed. No good. Right? What do we do? How do we say? What's the critical comparison? That's a good one, right? And the adrenal-ectomized moms and the non-adrenal-ectomized moms that weren't stressed have the same stress reactivity of their offspring, right? Both of those are great, right? Okay. Does that make sense? Do you understand? You asked the question? You got it? Okay. Yes. Is it another figure or about this one?
No, it's about.
Okay.
So is it like, another key component of the question that they're asking is the mom stress response due to the hormones released by the adrenal gland when the moms are stressed? Is that what impacts the stress response?
Well, I think what this is suggesting is that it's mom's hormones are affecting the baby, right? We don't, and this is the kind of crazy part that sounds maybe weird to say, but we really don't care about mom, right? What we care about is, and again, so notice that we stressed mom, but we took the hormones that could affect the baby away, right? And so that's the piece. So we don't know what happened to mom, and we really don't care, right? And I mean, I hate to be flippant about it, but I try to make it memorable as much as
So to
possible.
reword the question, we would just add, does prenatal stress affect adult stress reactivity, and is it dependent on adrenal hormones?
Yes. Okay. Other figures we'd like to go through? Now again, remember we've got social behavior, right? Both affiliation, sort of pro-social behavior and aggressive behavior. We have stress, and we have homeostasis, right? That's what's going to be on our exam. You would like to do that figure? Okay, we can do that. Talking about this one, right? Good. Okay. So, what are we interested in here? What are we trying to, now this is a complicated figure, right? So there's a lot of things that we're kind of interested in, of course, right? So what is the first thing? What are we interested in to start with?
We want to know how testosterone and lunar effect, how that affects or
So really what we're interested in, winning percentage, essentially, right? So we're interested in factors that affect wins, right? Winning conflicts. And you can imagine humans are very interested in winning things. Humans like winning. Everybody loves winning, right? It's way better than losing. And there's plenty of reasons why we can say it's way better than lose. It's way better for your endocrine system to win than lose, right? And that's, not unrelated to how you feel about winning and losing. People generally like to win and don't like to lose. So if we want to understand the probability, how can we increase the probability of winning in the future, right? So if we just take a rat, I think it might even be a hamster, but it's a rodent, some kind. I think it's a rat, maybe. So how often? Now again, so what kind of experiment is this? What's the experimental design basically? What is, if we want to test aggression, what's our model to test aggression? Resident intruder, right? Again, right? And this isn't just limited to the ones we've talked about. Like if you want to test anxiety, right? You test like Y maze, or there's always a kind of behavioral model to test these things. And so we are doing a resident intruder test, and we're asking, okay, how often does our resident win if you do nothing to him? If you do nothing to our resident, how often does he win? Make sure we're reading this correctly.
Like 25%.
Oh, 25%.
Yeah, a little bit over 25%. Not great, right? These are not great. All right. Now, how can we improve? How can we improve our friend's winning percentage? What's the first thing we can do for him? We can give him some testosterone. It's a great start. Now, how much does that raise his win percentage? Yeah, more. I like it. More than doubles. Over 50. More than doubles. The numbers, right? Numbers and more than doubles. Now,
Which one are we looking
testosterone in winning percentage.
Which chart are we looking at right now? The numbers that they're giving out.
Winners, left, top left. A, I guess. A, right? So now we're getting our guy up to about 58%-ish, maybe almost 60% wins. Now, if we really want to, right, get him winning, what do we need to give him?
Oh, yeah.
We're going to give him the testosterone, but we're going to allow him a win ahead of time, right? So he gets a win and he gets the testosterone injection, and then what is his win percentage? It's 100%. He's winning all the time. He's really full of himself at this point.
at? So we're really just looking at figure A.
Well, so far we've only talked about figure A,
Oh, okay.
right? So we're oddly enough about to talk about B and C. So if we look about that, and unfortunately there's not just a win by itself, but notice that there's going to be a slight challenge here, because just a win by itself is going to give you an androgen boost by itself. But we're going to even give them an even bigger boost. But so if we think about, all right, so we've got this change in winning percentage. B and C are really about the factors that affect that winning percentage, right? So we've got two measures of what here? In B and C, we have two.
Latency and attack.
What are those measures of?
Aggression.
Aggression, right? So we have two measures of aggression. Latency to attack, i.e. latency to initiate aggression, and how many aggressive events we have, right? So if you're just if you're like our lowest successful rat, how long are you waiting before you attack? I mean, obviously too long is the answer to that question, but it's about, 200 seconds, right? That's several minutes that you have another male in your tank. That's not so great, right? But you're like, no worries. Now, how long if you are really feeling yourself? I mean, you're on that rat as soon as it's in the tank, right? And how many times are you attacking it in what is this, like it's like a 10-minute period? How many times are you attacking it in that 10 minutes? About 35 times compared to our saline, 5, right? So, we've got a couple of things, right? How to change the probability of success and how the probability of success is in fact changed, right? The answer is, it doesn't, at least we're not measuring anything to do with submission, but the animals who got testosterone and testosterone in a win are much more aggressive. They produce much more aggressive behavior and they produce it faster. Yes?
So to make sure I'm reading it right, for figure B, the testosterone plus win, that means they're attacking the loser rat.
Not the loser. So this is a resident intruder paradigm. So what this means is that, so like if you're a rat, right, you live in your house, right, let's say you're a male rat, and then I'm going to put another male in there. You're the resident, they're the intruder, right? And so this is not the same rat that you got a defeat against. It's a new rat, okay? So, yes, so keep going. Sorry, I just wanted to clarify that.
So the resident is going to attack the intruder as soon as the intruder is in.
Yes, it's less than a minute after the intruder enters, right? I mean, we don't have the blown up scale bar, but I'm going to say that's probably less than a minute, maybe 20, 30 seconds. Maybe there's a moment of sniffing, and then an attack.
So the T means that the resident kind of like, I want to say chill, but like doesn't.
He's not as aggressive. He just the testosterone. Notice that if we're essentially starting, right, so this is our control, right? So that's 300 seconds, that's like 5 minutes, kind of, right? So we're dropping from 5 minutes to like 3 minutes maybe, right? We're waiting, and now here, it's less than a minute, right? So this is just, right, so the lower latency, this is kind of like, right, we did latency for like cup retrieval, like the bigger the number, the like less parental the animals are, right? So the smaller the number, right, anytime latency, so it's like mount latency, we looked at that, right? So if a male just like mounts within the first minute, right, he's more sexually motivated than a male that's like, you know, waits 5 minutes before he attempts to mount. Okay, yes? Well, so the question is all about increasing the, what we're interested in are factors that increase the probability of winning a contest, and why, right? So we're measuring the outcome and the behavior that supports that outcome. Another figure we could do quickly, or are you guys good for the day?
So, I'm guessing the takeaway is just, when you get the testosterone and you put it in, it decreases attack latency.
It decreases latency and increases the total number of attacks. So I probably, if, you know, to formulate it a little bit differently, I would say, because remember, our question is about factors that affect, right, winning percentage, and so testosterone and testosterone as a win, each of those in succession increase winning percentage as a result of making the animal more aggressive. Right? So androgens and a win are making the animal more aggressive, leading to a higher winning percentage, right? So you've ever seen like, you know, in sports where I was talking about like momentum, right? You know, they're talking about like a person loses their confidence, right? But if they hit a couple of shots or they win a couple of matches, then they get like this theoretical like momentum, right? This is kind of a biological equivalent of that sort of phenomenon, right? And like I said, if you know, it's like in the fight promotions, right? They always start the new fighters against people they're supposed to beat, right? So that they can get their confidence up. But when we really think about what that means, if you win a fight, you're more likely to win your next fight. If you win that fight, you're more likely to win your next fight, right? Yeah.
I have another figure. So it's the intact, the sham, and the chronology in stress response.
For this is the short-term.
Just short-term stress.
Yep. Okay. We got 5 minutes. We can do one more. Let's see. That's a little bit actually. There it is. Okay, this one. Okay. All right. So, what are we interested in here?
We're interested in how much an acute stressor affects the immune system.
Right, or really like a big picture, we're interested in how acute stress affects the immune response, I would say.
What is sham?
Sham is a surgery, so notice that surgery itself is stressful. So if I want to do an adrenalectomy, a sham adrenalectomy is that I'm going to make the same incision, I'm going to put them under anesthetic, I'm going to do everything exactly the same except for the last step where I remove the adrenal gland. And so what it's controlling for is the procedure, not the removal of the actual, it's a procedural control, right? And so especially important whenever you're doing things with stress, okay? So we're interested in acute stress and its effect on the immune system, and specifically whether or not that effect is mediated by the adrenal gland. Okay, great. So, first off, so remember, we're interested in the effect of acute stress. So, what is the effect of acute stress?
The acute stress would be the sham.
What is the stress? The stress is the blue lines. So every blue line is a stressor before one of these dead pathogens, right? So if we're interested in the effect of acute stress on immune function and whether that effect is mediated by the adrenal gland, so what is the effect of acute stress on immune function? It increases it, and we can see that by comparing.
The red line. with the
Right,
red.
so it increases it by how much?
0.2, I mean 2 point, what is 2%?
So you're getting an additional 2% increase in immune function by an acute stressor before, right? Okay, so remember, that's what we were asking, right? First off, part one is acute, how does acute stress increase or decrease, how does acute stress affect the immune response? It increases the immune response by about 2%, right? Now, remember our part two is this effect mediated by the adrenal gland? And so, what is the answer and how do we know?
Yes.
Yes, and we know because in the adrenalectomized animals, the acute stress did not increase the immune function over baseline, right? So remember our question was, what is the effect of acute stress on immune function? And is that effect mediated by the hormones of the adrenal gland? That was our question. To answer part one, we're looking at does acute stress increase immunity? That's panel A. We say, yes it does, by 2%. Then we ask, is it from the product of the adrenal gland? And we can say, yes it is, because now we have removed the effect of the acute stress by adrenalectomy, right?
So stress increased immune function by 2%, but without the adrenal gland, the immune function dropped to 3.
So I would say the immune response is no different, right? So notice the blue line is still stressed,
Okay,
right? The blue line is still stressed, but you're not getting the immune benefit, which tells us that it's some product of the adrenal gland, the thing we removed, that is responsible for the increase in immune function that we see in intact and in sham. I mean, and again, I'm just using the intact one, but you could do the exact same thing for the sham one.
Can I say, you know, how the significant difference? The significant difference. I say, the significant difference.
Yeah, that's fine. Yeah, no difference, no significant difference, right? Roughly the same, right? All those are fine. Okay. No, it's good. So a sham is a control for the surgery. So notice that the adrenal glands are on the top of the kidney, and so in order to remove them, I have to cut the animal open, I have to move its insides around, I have to find the adrenal glands, I have to chop them out, then I have to sew the animal back up, then it has to recover. That experience is stressful. So in a sham, I'm going to anesthetize the animal, I'm going to cut them up, I'm going to move their internal organs around so I can find the adrenal glands. I'm not going to remove them. I'm going to sew the animal back up and let it recover, right?
Okay,
Are you going to ask me what a sham is? I know, I've got that same question like 4 times today, so that's okay. Yeah.
Right, okay, so we're comparing the intact and the adrenal gland, so the sham is controlled, we're comparing the intact and the adrenal gland to the sham.
Yeah, so the sham is a technical control, right? And the important thing, and the point that I was trying to emphasize here, right, is that what's the magnitude of the difference here? It's about 2%-ish, right? 2%. Now, it's likely that the stress of surgery, to me, it looks like it knocked the overall immune response down a little bit, right? We talked about that a little bit there in the time, but the important part is that if these two things were different in the effect, it would say that our technique didn't work. So I'm trying to say you can compare to the sham, and that's what I kind of made that point. You can compare it to the sham, you can compare it to intact, right? It really, because these two things are the same, they're both controls, right?
Does that mean adrenal-ectomized patients would not heal?
It means that they would not get a benefit from an acute stressor preceding, right, an infection, right? So basically what it means is an adrenal-ectomized patient is going to have a huge amount, if you're talking about a human, right, or in a long-term study, then you've got to have glucocorticoids and you've got to have, the stress response is just screwed at that point. So they would not have a stress response, right? Basically, no epinephrine, norepinephrine, and no cortisol. So they would have lots of problems, right? In addition to this, but so notice that the question that we're asking is just basically, does acute stress increase or decrease immune function? Because what does chronic stress do to the immune function? Chronic stress? Yes. Chronic stress depresses immune function. Acute stress increases. And we know acute stress increases because if we give an acute stressor before exposure to a pathogen, right, we get a 2% increase in our sort of all-cause measure of immunity, right, like T cell counts and all this good stuff.
So, I think we'll
What is the takeaway message? Yeah, you can't get away that easy. So what's the takeaway? So what was all right, so again, what was our question? Does acute stress or increase immune function, improve, increase, change immune function, and
is it controlled by the adrenal?
so the take-home message is yes, it is. It does increase immune function, and that increase is mediated by the products of the adrenal gland. Right? Okay.
Wait, I have a question. I'm sorry, it's just one question. It's not about a figure. I just need you to describe one label in the figure.
Okay. What label?
What we did today in the aldosterone MR, the
Yeah.
scrambled, I never really understood what the scrambled was.
Okay, so an antisense oligonucleotide, right? So you have some mRNA, right? This is your mRNA, right? It's like got like a bunch of like, right? GC or GGC. See, just for simplicity's sake, I'm just going to do GGCC, right? So there's some specific sequence that is the RNA, the mRNA. An oligosense oligonucleotide is a specific sequence to bind to this, right? So that is antisense to this. This is sense, this is antisense. Notice that our antisense oligonucleotide binds to this and forms a double strand, and then they chop it up. A scrambled one might be something like this. All of the nucleotides scrambled up so that they don't bind the mRNA in question.
So it was just a control for the blocker?
It was a control for the technique, yes.
I'm still, you said what now? So, what does MR stand for?
Mineralocorticoid receptor. It's the aldosterone receptor.
So when you say it's scrambled, that means the receptor isn't made.
In the antisense, right? In the antisense, the receptor is not made. In the scrambled, the receptor is made.
So what's the scrambled?
So it's scrambled to control for injecting single-stranded RNA into the animal, right? It's like a sham, since we just talked about that, right? It controls for the procedure itself.
Okay.
Right? So if I said, like, and this is particularly, right, it's not a trivial thing because a lot of times, like, the procedure actually affects the result. And so it's the result of what you did to the animal, not the manipulation that you were trying for, right? So sticking a needle into the brain of, or the kidney or wherever of an animal and injecting like a nucleotide is what you're controlling for, right? It make sense?
It is.
Okay.
Did you have a fight today?
Hmm?
Did you have a fight?
Did I have a fight today? I went to training. I'll blow it up. Yeah. Yeah, it's all scratched up. It's okay. Yes.
Close to what?
Grad project.
Oh,
Oh, what? When is the grad project due?
Like finals week.
Okay. I didn't even know you did grad project.
Yeah, I might have. I don't know if I took it out. I don't know if let me see if I took it out.
Huh?
Yeah.
He said he's gonna post it soon. I don't know when. I need more grades on that. We need to get me more grades in there.
Oh, yeah. Okay. She's in class.
It's okay.
Oh, wait. Yeah, no, I think I had you somewhere.